biology has evolved to evade the entropy. Like if it doesn't, you will age immediately. You will not survive.
Welcome, everybody. This is Buck Joffrey with Longevity Roadmap. And today, really interesting conversation with Dr. Derya Unutmaz. This is a very interesting guy who's really known for his groundbreaking research on the immune system and T cells, how they relate to aging, and a lot of the things that we've sort of referenced in the past. When you listen to this conversation, I'd say the first half of this interview really dives into the nuts and bolts of the role of T cells and aging. And I want to warn you up front that gets into gets into the weeds. And so if you're not super excited or that interested in a lot of the basic science, you may want to forward that. But what I don't want you to do is to miss the last half of the interview, which is really interesting conversation on the concept of aging in general, and his particular view that this concept of longevity, escape velocity, and ultimately the ability to completely reverse the aging process is not only possible but inevitable in as short as 20 years from now. Anyway, don't miss it. Really interesting conversation, really smart and interesting guy who thinks not only at the level of behind the bench in the lab, but also at a very, very big picture on aging. When we come back, we'll have that interview with Dr. Derya Unutmaz.
Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to Longevity Roadmap, everyone. And today I have had the pleasure of having Dr. Derya Unutmaz, a distinguished immunologist and professor at the Jackson Laboratory for Genomic Medicine. His research is centered on the role of T cells in regulating immunity, aging, chronic disease. He's recognized as a pioneer in the field of immune resilience and is advancing innovative strategies to promote health and combat disease. Welcome. How are you? Thank you so much.
Thank you very much. Great to be here.
Well, I think just to begin, I want to make sure that we have a little bit of a reminder for our folks as we start this conversation on T cells. Just generally speaking, sort of a reminder on the role of the T cells within the immune system.
Well, I've been studying T cells for the past 35 years, so I might be a little bit biased about their role. But I do think, I think most immunologists would agree, they have a very critical role because they're sort of like the generals of the immune system. They control, regulate variety of things. In fact, we know this from HIV infections. HIV targets a subset of T cells called CD4 T cells. These are sort of they used to be called helper cells, but they actually secrete cytokines. They help macrophages, B cells, other cytotoxic cells. And so when virus depletes these cells, you get severe immunodeficiency and you die. So it's clear that they're very, very important. And I think one thing that's not maybe as much appreciated, not maybe for your audience, but in general, they are also very dangerous cells. So they have to self-regulate. In fact, we studied a subset of these cells called regulatory T cells. I call them the bureaucrats of the immune system. We made some discoveries about that, and their function is to actually suppress and control the other subset of T cells because they are very dangerous. And probably play a role in many chronic inflammatory diseases, some of the autoimmune diseases. They're important
Yeah.
for
Yeah.
good, bad, and maybe the ugly.
So when we think about them as sort of both defenders and regulators of the immune system, how do they change as we age?
Yeah, that's a great question. I think immune aging is very important and something that I'm obviously very interested in, because it impacts a lot of the aging related to diseases. And I think if you want to reverse aging eventually, or extend human lifespan, we need to focus on the immune system. And T cells are critical there because these are long-lived cells. For example, obviously macrophages, neutrophils, other immune cells also play a very important role, but they can constantly regenerate, and new ones come out from the bone marrow and other places. But T cells have a memory, so they can remember things, and they can get exhausted, they can get senescent, and they just hang out there. And so that becomes a major problem during aging, because especially there's not much renewal going on. As you know, the T cells come out from the thymus as sort of naive cells, but that process stops probably during teenage years. So you have very, very few cells emigrating into the periphery from the thymus. So the T cells have to be self-sufficient in the periphery. And in fact, there is a naive pool of cells. We call them naive because they haven't seen their antigen yet. And those cells are both very long-lived, and they have a half-life of probably 6-7 years, but they can also divide occasionally, so they can maintain that naive pool. In fact, one of the things that's very predictive during old age probably for all mortality, is this reduction in naive T cell population. So if your naive T cells are going down and your effector cells or memory cells are just expanding, then you have a very high risk of dying from pretty much any condition, because they have to constantly renew. Not only when you're exposed to new infections, but also from the regulatory aspects, they have to constantly check the ones that have become effector cells. So when you start losing those populations, you are more susceptible to inflammatory diseases, cancer, of course, infections. I mean, we saw that during the pandemic. Most people who died were over the age of 60, 65, and that had to do with their immune system. There's a very tight link between the immune system and the aging process.
So we call that inflammaging often, right? This concept
right.
of
Yeah.
So inflammaging, do you think largely is a T cell phenomena then?
It's not just T cell phenomena. I think actually most of the mediators of inflammaging, like pro-inflammatory cytokines, TNF, IL-6, these are typical things that everybody knows, are actually secreted from macrophages. So I think macrophages are a major problem, both from the aspect of they secrete these pro-inflammatory cytokines, but also they don't do their function as garbage collectors, I would say, because macrophages actually clean the environment of that cells or extracellular matrix. And so, once they go into the tissues and become sort of these semi-long-lived resident senescent cells, they are a major source of these cytokines. But having said that, again, the commands come from the T cells. So if the T cells are not regulating those and constantly sending signals that something going wrong, and also the other cell types in the environment do contribute as well, because as other cells get senescent, they also start to produce inflammatory cytokines, and they will activate the macrophages. But the T cells play an important role there, just to sort of calm them down. And I think even if they are not directly contributing to the sort of the inflammatory cytokine milieu, they are definitely contributing by not controlling these events. But there are subset of T cells that do directly contribute. There's a subset called Th17 cells, which produce this inflammatory cytokine called IL-17, and there's a subset that produce interferon gamma. So those actually directly do contribute to the inflammation process as well.
Going back to this idea that in a way there's an inevitability of losing these T cells because of aging. You mentioned that these originate from the thymus, and the thymus, of course, starts to become degenerates over time pretty quickly. There has been a fair amount of research on the concept of thymus regeneration. I'm curious on your take on that. I know we've had people on the show talk about various protocols involving metformin, growth hormone, things like that. What is your view on these?
I think it should be possible to regenerate thymus, or at least sustain it a bit longer. It's not just the thymus; it's also some critical cytokines. For example, IL-7, which is a major survival factor for these naive cells. In fact, as cells differentiate, they lose the IL-7 receptor. So what IL-7 does is that it sends these signals through mTOR pathway, just keep the cells alive for very, very long periods of time in a sort of a pristine manner. There are a couple other cytokines like that too. The regeneration has to happen within the thymic stroma, because there are specialized cell types there. Some of them are dendritic cells. So thymus is kind of like education area, right? So like a university. So the thymus has to very carefully select those that shouldn't graduate, because those would be dangerous guys. They would be autoreactive T cells. So I am not sure that could be achieved simply by taking things like metformin or growth hormone, because it's a very complex transcriptional molecular mechanism that's going on there. There are certain FOX genes, for example. You have to sort of regenerate and regulate other cells, not just, of course, you have to have these stem cells from the bone marrow coming into the thymus as sort of what we call them double negative cells. They're precursors of CD4 and CD8 T cells. So it is a fairly complex issue. But having said that, I think, in various mouse experiments, if you transplant thymic to an old mice, you do generate new T cells. So you still have the machinery there, but it does get involuted. So I think, one important question is, what is the evolutionary basis for that? Like, why is it that thymus doesn't survive longer? And because we have to ask these questions, there could be a cost of that, right? So having too many naive cells coming out all the time, it may not be a great thing as well, because you have limited niches. You do need to maintain a significant memory effector population. This is different than B cells, for example, because B cells are constantly being made. They're much more dispensable. You have very few long-lived memory B cells that make antibodies. But I think it's, at least in a limited manner, we probably should try to regenerate the thymus. I don't know whether that's going to require genetic engineering or other more sophisticated methods that should definitely help for the aging process of the immune cells. But we have to think it in combination with also getting rid of these sort of the troublemakers, effector cells that are not really senescent. They're terminally differentiated. They are very pro-inflammatory. They just stick around. So if they stick around, you can't have the new cells coming out. They're not going to leave their niches, and that's a major problem. It's a major problem in other organs like skin and other places as well. We got to somehow figure out to get rid of them.
So we know that obviously T cells play a big role in fighting infections. They have a role in, I guess, preventing or even accelerating chronic diseases, right? Heart disease, diabetes, Alzheimer's. Do you see any potential opportunities, sort of at an organ specific or disease specific level?
Yes, definitely they play an important role in those inflammatory conditions. But I don't think we have to discriminate that from an autoimmune condition, right? So in an autoimmune disease like type 1 diabetes, there are T cells that specifically recognize individual peptides or antigens, just like they recognize a virus, but just happens to be a self peptide. So there, you kind of know the targets and you can try to interfere with them. But in a chronic inflammation, there is, as far as I know, there isn't a sort of a self trigger like a molecule that these cells are. I mean, with very few exceptions, it's mostly the cells feel that there is a constant threat. Whether that threat is disruption of the microbiome or hemostatic mechanisms or not sufficient regulation, the cells are under constant alert. And that's actually the pillar of immune system, that it acts according to the dangers. So we call it the danger theory. So if there's no danger, the immune system doesn't really care. But at the same time, I think that's also could be a way to interfere with that in a more generic way, not just looking at specific T cells, but how can we sort of lower that trigger that causes, like Th17 cells or Th1 cells to produce these cytokines, stimulate macrophages, and so on and so forth. I will point out the microbiome a lot because, some of our research and many others show that, that is a major source of this trigger.
Yeah, talk a little bit about that, a little bit more about that. We're curious because, we have on this show talked much about the role of the microbiome. And, so how much do T cells interact with the microbiome? Could you explain why gut health ultimately seems so tied to aging?
It's definitely very tied to aging, and not only aging, but I think it's tied to many of these chronic conditions, quite a few of the autoimmune diseases as well, because it's very tightly linked to the immune system, as well as the nervous system, actually. And the reason for that, we're beginning to understand. Imagine you have these, I don't know, close to 100 trillion foreign organisms that you have to make peace treaty with, right? So they live in your gut. Quite a few of them are very useful for us. They digest our food. They produce all these metabolites that are necessary for us. Like riboflavin, vitamin B2 is purely made by the bacteria. Yeah. So the immune system has a challenge. What do I do with that, right? So, and then some of them are potentially dangerous bacteria. They could be pathogenic if they find the opportunity. So there's constant communication. There's constant treaties being made and broken, and occasionally the immune system responds to it. But at the same time, if you look at it from the perspective of the bacteria there, they want to live there. So they have adapted evolutionarily to control the immune system. And so they produce lots of metabolites, which in turn actually regulate the immune system. But that turns out to be actually useful because, under chronic inflammatory condition, you want that. You want those bacteria. I'll give you an example. Those bacteria that produce butyrate or short-chain fatty acids. So it turns out that butyrate actually could be a negative regulator of certain pro-inflammatory immune cells, especially T cells, or tryptophan metabolites. They could also act as sort of suppressors. So both of these systems have kind of, in a synergistic manner or opportunistic manner, they adapted. Okay, we can use that to regulate excessive immune function, and then, okay, you can use this so that we don't attack you, kind of a thing. But when that breaks down, when what we call dysbiosis happen, when there's sort of the let's call them the more pathogenic bacteria that live there increase. And then actually we start to learn the sort of the phyla of these, like Bacteroides and Firmicutes and things like that. So there's like thousands of strains, and some of them starts to break that balance, and so they don't produce enough butyrate. So if they don't produce enough butyrate, then the immune system becomes a bit more active. Then they produce other things that will trigger more inflammatory reactions. And then, all of that is also linked to your diet, because you can actually manipulate the bacteria using changing your diet habits. Certain bacteria likes meat, others like, vegetables and so on. So there's still lots of gaps in that because they produce thousands of metabolites. We only know a small portion of that. And I think as we learn more, the nice thing is that this is actionable, right? So if we find that certain bacteria that's lacking butyrate, well, we might be able to replace that, either the bacteria or the metabolite. So I'm trying to really summarize in a very generic way, but there's obviously lots of details there.
Sure. In terms of therapies, so in cancer therapy, we've come to a point where we have learned how to reprogram patients' own immune cells, in certain cases called, CAR T cells. And a big breakthrough. Those cells can essentially specifically attack tumors. Can you explain sort of the process of engineering immune cells and how, if we can use it against cancer, it seems like there could be a broader application to aging. Just your thoughts on that?
Sure. Yeah, that's a great question, and something that I'm very, very interested in. We actually been working on that topic for quite some time. In fact, I think I was one of the first to develop what we call lentiviral vectors back in 1998. It's been a long time now. So what lentiviral vector means is that it's actually a sub-portion of HIV. So I used to work with HIV, and we realized, as well as many other labs, this virus knows the T cells very well, and it actually integrates into the genome, so it can deliver the cargo, whatever the gene you want to send, and become part of the cell. And so we and others engineered these vectors where we took out sort of the parts of the virus that makes it replication incompetent. So it cannot divide, but it can still go in and integrate into the genome of the cell and then deliver the cargo. So that's the principle of these CAR T cells. After we used it many, many times for genetically engineering T cells with CRISPR, with expression transcription factors, and all kinds of things for research purposes. And the idea was that, T cells have specificity. They need to recognize something, which is usually a small peptide present in the virus or viral infected cells that is presented by these MHC molecules. This is very different than antibodies, right? So the antibodies will bind directly to the antigen and they will recognize the conformation. But the problem with the cancer is that these are also self cells, right? So almost all of the antigens in a cancer are selected against in the thymus. The T cells don't want to respond to that. So what do? You can find what we call neoantigens, and people are doing that with mRNA approaches. There are mutated antigens like p53 gene, which if you can identify that, and you might be able to generate a T cell that will only recognize that peptide. But this is going to be different for every patient. So it's not a trivial thing to do. The alternative is that what if the T cell can recognize something on the surface of the cancer cell that might be shared with normal cells, but it's either more on the cancer cell or it won't cause a major problem, even if the cells react to the normal tissue. An example of that, like in breast cancer, is called HER2. HER2 is an antigen on the surface of these cells. Normal breast cells have HER2, but a specific set of cancers have much upregulated version of HER2. because it signals the cells, the cancer cells to divide. So if you can engineer a chimeric molecule where outside is an antibody that binds to HER2 molecule, so it will only recognize HER2 molecule, and then the inside is a signaling domain, which mimics what would happen if the T cell were to activate it by an antigen through their T cell receptor. So as a couple of modules of the signaling domains. So, and then you deliver that with a lentivector, what we call the CAR, chimeric antigen receptor. It's because it's a chimeric molecule. It's expressed on the cell surface. So the outside is an antibody that recognizes this antigen on the surface of the cancer. It could be CD19 on a B-cell lymphoma or HER2 or whatever is in the cancer surface that you think you can design it to. And once that binds to that antigen, like the HER2 molecule, that will send a signal to the T cell, say, okay, get activated and kill that guy, because the T cell doesn't know whether that's a viral infect or not. So that's the principle, and it's been tremendously successful in cancer. They're not easy to make, so you have to generate them in the lab from the patients and every time and so on. Although we're figuring out ways to do it more kind of like off the shelf manner. So then people said, okay, well, what if we use that, exactly coming to your point, use that against these so-called senescent cells, these cells that are supposed to retire, but they don't. And we're having difficulty getting rid of them. And several papers were published as a proof of concept. If you can find an antigen, again, it has to be on the cell surface, that is more so on a senescent cell, you can generate a CAR molecule that will recognize it, and that you can actually eliminate it. People have done it for the heart muscle, for skin cells. The challenge there, again, is to find something semi-specific, right? So we're actually looking for that too. Many others are doing it as well. That's somewhat upregulated on senescent cells. So you don't want to damage the normal population. There is one variant to that, which might be a better approach in engineering for the aging or for senescence. Actually, it could also be used for cancer. So it's a variant of what I mentioned earlier, that it's easier to find peptides that are different because even in senescent cells, you're going to have mutated genes, right? So if you can find a peptide that is somewhat mutated or more so in a senescent cell, then one can generate an engineered TCR, T cell receptor, rather than trying to generate T cells in each individual, because you may not have the right T cell receptor to recognize that. And because they're selected against, it might be very difficult. But we could generate a T cell receptor that will only recognize that peptide and then engineer that back into the T cells. And we and others have done it. Actually, in melanomas, this was one of the first trials. People identified specific melanoma peptides from an antigen called GP120. And so you can make these T cells, and it's like a clonal army. They all have the same T cell receptor. And you can even knock out the endogenous T cell receptor, and they will go and kill these cells. So again, for the cancer, people are trying to do that for these neoantigens and then generate these TCRs, because again, that solves a major problem that you don't have to start from scratch for every donor. You have already off the shelf engineered T cells ready to wait there. So that's another alternative approach.
Seems like a broad approach for, just in general. We're pretty far away from that. Just a broad approach to just the aging process or that kind of thing. Of course, we talk a lot about longevity and escape velocity and all these concepts here. Do you see any of these technologies coming to fruition anytime soon in terms of actually sort of battling the inevitable process of aging?
Absolutely. I'm more optimistic than ever because of AI. I think if AI wasn't at this point, I would be less hopeful that we will solve this in the next, I don't know, reasonable time, like 20 years or 30 years. It would have taken us much, much longer. But actually, my timeline, which I predicted 20 years ago, haven't changed. Around the 2040s, 2045s, we should be able to completely regenerate the biological systems such that, it's not stopping or slowing aging, but reversing aging. And
Do
I
you
think
mean the immune system in particular, or just
immune system, but I think as a whole, every organ might need to be dealt with separately. Some could be more generic, some could be very focused, because different organs have different needs. Like, the brain or the heart muscle cells don't regenerate very much. So we're going to have to fix the cells that are already there. And of course, the brain neurons, you don't want to get rid of them. Whereas the skin, you can get rid of all the senescent cells and then get newly generated stem cells, clean up the niches. The reason I'm so confident about this is that this is what biology does anyway. There's no a physical limit that you have to age and the system has to break down.
let's drill down on that a little bit, because I think it's an interesting concept. When you say it's what biology does anyway, you're talking about the repair mechanisms, the things that when we're young, we automatically do, right?
That's right. I mean, take, for example, the immune system, right? So we have a population of these naive cells in the periphery that constantly repopulate. And, as you get a lot of effector cells, they're removed. I mean, so the cells expand thousands of folds. And then once their job is done, 99% of them are killed. They go undergo apoptosis and whatnot. And then you have new cells coming in. So this is happening all the time. The moment you're born, this process starts. The same thing happening in your skin. The cells divide certain time. They reach what's called the Hayflick limit. Their telomerases are shortened. They die. New cells come in. The cells in your gut, they renew almost every day. There's a stem cell at the very bottom of what we call the crypts. That's the mother stem cells that divides, I don't know, once every few months. The next one divides it once every few weeks, and then so on. So they constantly replace the gut epithelium, on a daily basis or every few days. So the point I'm trying to make is that this process is happening. Or you have, DNA repair mechanisms. Our DNA is under constant bombardment with mutations. But, we have genes that specifically focus on that. They repair it. We have epigenome regulation, right? So epigenetic changes are very important because some of them can be irreversible. Not irreversible, but they push the cells toward terminal stages. But you have mechanisms that control that. So all of that breaks down as you age. We don't know why that happens. It's, again, an evolutionary reason. A blue whale lives hundreds of years. Our biology is not very different. Why is it that they can keep living and we can't, for example? Or a mouse lives only 2-3 years, and they're also mammalian. And actually, we do a lot of experiments with mice because they're similar to us. But it's very complex because there are millions and billions of parts that are interacting in a very network fashion. That's where AI comes in. So AI can really see all that information. We just need to collect a lot more data and create these patterns. You can say, well, here's what you need to interfere with. And let me screen 1 billion compounds, by the way, and then find you the exact molecule that will act there. Or this is the peptide you need to develop your CAR T cells to. And this is the time that you need to deliver it, and so on. So that will tremendously accelerate the whole process. And I'm very confident we're going to get a handle to this.
So I'm curious on your timeline. Where do you get the mid 2040 projection? I mean, obviously, we have AI now. We know that accelerates things in terms of, a lot of things you're talking about. But tell us what you think about, like the big picture here. Where's the target? I mean, obviously the immune system is a big part of it, but it seems to me that if you're talking about, a sort of master switch or a master mechanism, do you think we've identified that? Do you know what I mean?
Yeah.
So there's a couple different ways to look at this. Talking to Aubrey de Grey, for example, he talks about longevity escape velocity, and his take is essentially that we can essentially get to the point where we can reverse or fix the things that are wrong with us, and then live long enough so that the next time we have a problem, we have a solution for that as well. That's one way to look at it. The other is a master switch, which we've discovered that this is what goes wrong, and this is what ultimately, creates this cascade that we can stop. So what is your view on these kind of ways of approaching aging, and what do you you don't have a crystal ball, but where do you think that answer is going to be?
Yeah, so it's probably two questions. They're linked. I'll start from the second one. I don't think there is a master switch. It's not. Let me put it this way: there's no such thing as free lunch, right? So it's not going to be that straightforward, because there's a lot of noise. There's a lot of stochastic events that happen, both at the gene level, on the protein level, on cellular level. So it doesn't make sense for evolution to control such a multicellular organism with one switch, because if something goes wrong there, you're gone.
Let me rephrase it, because you brought up the example of the whales and other organisms that live much longer than us. Whales, of course, genetically, they're not that different from us. And we know that, for example, they have a lot more sirtuin genes. They express a lot more of these, specific aging related diseases. And when I think about a switch, maybe it's not one, but maybe it's a dozen big picture genes that are controlling the speed at which we age.
Yeah.
Is that?
Yeah. No, that's probably more reasonable. In fact, if you look at the developmental biology, right? So we can recreate ourselves from a single cell, right? So you can actually you can take a skin cell, reverse it back to what we call iPS cells, pluripotent stem cells, and you can regenerate everything that you have from that already programmed cells that's regenerated. So that suggests that it can't be thousands of switches, as you said. There's probably, in fact, even during developmental stages, there's things called Hox genes, for example. So this is a family of genes that there are kind of the orchestrator, right? So they give a lot of commands, and then they create cascade events and feedback loops and so on and so forth. So a lot of those things we also know what they are. Reversing the cell back to an iPS, so you just need like 3 or 4 transcription factors. So we know those genes regulate a lot of other genes. They can reverse or reset the epigenome and so on. So my point was that there might be somewhat different sort of the tuners depending on the organ. Like the brain could be a bit different than how the immune system is regulated or ages or regenerated versus the skin. But again, if you look at it within that system, you're probably right. I don't think there's too many of those things. The issue is that that's why I said there's no such thing as free lunch, because all of these things are also very, very tightly regulated, right? So it's easy to make something immortal, right? Cancer. So those cells don't die. We can have immortality, but that's not what we want. So how do we make sure that you don't overdo, like going back to example of the thymus? So we probably don't want too many naive cells. we want just enough. How do we regulate that? How do we make sure that there's enough of those under certain conditions? And then, it will be different in people with different genetic backgrounds. You know, not everybody ages the same speed. Some people live much longer. Women and men have very different immune systems. We beginning to understand that. I mean, women live always 5-6 years longer than men, for example, and we think part of that, or maybe most of it, is because of the immune system differences. So it needs to be adapted to those environments. But again, I think the key point here is not intractable problem. It is a tractable problem. And now going back to your other question, why I think this will be feasible in the next 15 years or maximum 20 years, reversing. But I think we'll reach the longevity escape velocity much earlier. My estimate is within 10 years, if not earlier. Actually, we began to achieve that. I always give the example of GLP-1 drugs. Some estimates is that if everybody who needed GLP-1 used it, you probably extend lifespan by between 5 to 10 years, maybe even longer. So you already added an escape velocity there. So the time is really based on how the technology is accelerating or evolving. This is not just AI. Of course, AI just came in the right perfect moment. But before that, we developed these omics-based assays. When I say omics, doing RNA-seq, which I think your audience would know. Basically, RNA-seq is to determine the gene expression in every cell. There's thousands of genes that are being turned on and off. And so if you want to understand what's different in a senescent cell and normal cell, we need to look at all those thousands of different genes. Now we're able to do that on a single cell level. For millions of cells, we can say which cell has what gene. And now because of the AI and the bioinformatic tools we have, we can also analyze those. The same thing is true for genome analysis, right? We've sequenced thousands or hundreds of thousands of genomes. We know all of the mutations that happen, the SNPs, and how the epigenome changes, and we know a lot about the microbiome. We've sequenced thousands of these bacteria. We know a lot about the metabolites. We've identified thousands of metabolites. It's not finished yet, but the point I'm trying to make is, the technology has advanced to a point where it's just a matter of generating more data so we have a complete picture. Because there are trillion parts, we already know 100 billion, but to get the next 900 billion is not going to take us another 50 years because the technology is to a point where we could just do this in mass scales. We are even able to now beginning to look at individual metabolism that's happening in a single cell. What's going on in your mitochondria and whatnot. And so, but the bottleneck then was, how do you analyze that data? How do you put it together? And that's where the AI came in. In fact, when I made this prediction 20 years ago, I said, we're going to need AI because otherwise there's no way we can make sense out of this data. And it just happened. And AI is getting better and better. I do a lot of tests. I was testing, GPT-5 from OpenAI before they released it. I'll give you one example. We were working on this disease called chronic fatigue syndrome. We had collected millions of bits of data, metabolites, immune system, and so on, microbiome. And, it took us a long time to analyze that, like over a year period, we had to analyze it. And so I took a part of that, like the metabolite part. We had 1,300 different metabolites from 200 patients and healthy controls. Just uploaded that to GPT-5. It took about 3 minutes. It came out with almost exactly the same analysis, and then also showed things that we had missed. We have spent like probably months just doing that. So that now accelerated everything in an incredible manner. So at this point, all we need is a lot of data. And that's why, I'm thinking the next 5, maximum 10 years is going to be spent mostly collecting this missing links, the data set. After that, once we have that data, finding cures or finding solutions will probably happen in a matter of days or weeks. We're not going to spend years looking for that. Now, there's still one bottleneck that we have to solve, and that's the clinical trials. Because you can say, well, okay, great, you found the drug, but you're going to test it on some old person and wait. Yeah, okay. That's not going to work out very well, right? And this is true for other diseases too, right? It takes so long to do clinical trials. But there's a solution for that too. And the solution is what we call digital twin. So, we will come to a point where once you have a complete picture, all of the parts of an individual, in fact, we can even personalize that. You upload that to AI. The AI has a biological replica of you. And so you can actually run the experiment in silico, in the computer. I can say, okay, what happens if I give this drug? What will happen to the metabolite in the microbiome that will then act on this cell? Or if I kill these senescent cells, run me the experiment, run me the scenarios. And so I think this is probably not far from 10 years. It might be sooner because all we need is a lot of compute. Probably need 1,000-fold more compute than we have now. If you can run that simulation, then you can just run the clinical trial in silico. We don't have to wait 10 years for the finding. Or we can really restrict it to a very small subset of people for the final validation. So I think this will revolutionize drug discovery. We'll be able to find drugs or solutions or therapeutics for pretty much every disease imaginable. And then, of course, the mother of all disease is aging. And that might take, maybe a few more years after that. We will definitely get there. I think people are not used to this because they're not thinking exponentially. They are thinking that in the next 10 years, we're going to have an advances like the last 10 years or like 20 years. No, next 10 years is going to be more advances than the past 100 years. So imagine the life in 1925 versus now. That's how different it's going to be between now and 10 years. In 20 years,
and that's mostly just because of the compute element, right? The
absolutely
AI.
Because it's doubling every 3-4 months in its capacity and compute and intelligence. I mean, we see this. We're living through it. I've been using GPT since it first came out, less than 3 years ago, and it's unbelievable the advance that we had during this period, and it's accelerating. Actually, it's getting faster and faster. So that's why I'm very, very excited. I think it's just a matter of time. Of course, we do need quite a bit of resources. We need to invest into this. People are spending hundreds of billions of dollars to data centers, but let's spend some to collecting this biological data.
There is, it seems like there is a lot of interest in the aging, especially, you've got a lot of billionaires who are getting older, right?
Yeah, actually, Putin and the Chinese premier, they I don't know if you saw that.
I did. Yeah, they were chatting about it, right?
And they're chatting about it. Yeah, now we can probably become immortal. Not sure it's a great idea for them, but they're going to put a lot of resources to this.
Well, right, and then private money as well. I mean, you've got, a lot of these, these people who made their money in .coms, and they're getting older. And are you seeing, by the way, just out of curiosity, I mean, like, are you seeing a big difference from, like, say 20, 30 years ago in terms of private investments in this from, large donors?
Absolutely. Even compared to 10 years ago.
Yeah.
There's massive. I mean, there were some pioneers, but people, because, even Sam Altman invested in an aging company. Google has, of course, Amazon has their own. And so because the tech guys now see the light at the end of the tunnel, that this is now feasible, because before it was kind of like a science fiction, very few people believed in this. There's tremendous interest. Well, one thing that needs to change in terms of the the policy, the government policy, which I think is also beginning to change, aging was not and still is not recognized as a disease. It's it's seen as something natural. Like I can't write a grant to NIH and say the purpose of this grant is to reverse aging process. That's not going to get accepted. That's why I'm very adamant against this notion of what's called healthy aging, because this is sort of used as, okay, don't touch the aging itself. We should all age and we should all die, but let's just live a few more years of healthy life. Let's fix that. This includes a lot of scientists in the field. Healthy aging is an oxymoron. There's no such thing as healthy aging. Aging means your system is starting to get bad and bad and bad, and that's why you have 90% disease happen in the aging, and close to 50 million people die directly or indirectly because of aging. It's a 100% mortality rate, right? So that's not a very healthy thing.
Bring up an interesting idea, which I kind of doing these conversations, I hear a lot of very smart people like yourself on, and let's say David Sinclair or those on one side who really are really bullish on this concept of age reversal, significantly lengthening lifespan beyond not just to 5-10 years, but 100 years, that kind of thing. And then there are others who are very, very smart. I mean, Charles Brenner comes to mind, who you probably know if you're following this. And Charles is a very...
We don't get along very well.
Probably not. I'm sure he's attacked you on Twitter or whatever. Where do really, really smart people on this, what fundamental question are they not agreeing on?
Honestly, I don't understand. First of all, in my opinion, anyone who doesn't believe that aging can be reversed shouldn't be in this field. Why are you working in this field? Like, what is your point? Because that creates a sort of a perception that you have other agendas, right? So let me just create something like NAD precursor, and then I'll start a company and then sell it to people so they can live an extra 6 months or a year or something like that. This is not the reason to work on an aging process, because they don't have a scientific rationale to say that this is not possible. What these people say, this is too complicated. Yeah, it's too complicated. 100 years ago, it was extremely complicated to go to space, right? Like, who would have imagined that now we're trying to send a starship to Mars? Or who would have imagined a computer or AI or internet or any of these things that we have now? These are so complicated things. But that's why we do what we do. So we can break the complicated things apart. And now with the tools like AI, put them together. Because if you say, okay, well, there's a physical law that you cannot pass this law. Like the light will not travel faster than 300 whatever thousand kilometers per second. That's physical law, right? There might even be ways to figure that out, but there is no such law. There's no such biological law. People make the point of, oh, well, there's entropy. There's the second law of thermodynamics. Well, biology has evolved to evade the entropy, right? So it does for years. Like if it doesn't, you will age immediately. You will not survive. I mean, there's a disease called progeria, right? So it's a genetic mutation in one single gene that has to do with the nuclear structure and so on. And these kids, they age 100 years in a matter of a few years, and they die by the age of 8 or 10. So everything gets accelerated. And again, there are organisms that live. There are crabs. There are certain crabs that have lived 300-400 years. They probably could live thousands of years. They just don't age. There are other organisms that constantly, you cut their head, they regenerate a new head. Like certain worms, or their arms, or whatever. So biology has that capability to regenerate, to fix, to to heal, to eliminate the toxic aspects of things. It's just that it stops. Again, it's an evolutionary selection. Why do whales live so much longer? Because there are very few threats to whales, right? Who's going to kill a whale? So they're the largest animals, so they can afford, and they have plenty of food. They have the whole sea for themselves, and so they could keep living. Their biology didn't get selected against that. The short answer is that I don't understand it. I don't understand what is their argument against this. So even if it was very, very, very difficult, your job as a scientist should be to figure that out. That's why you're working in the aging field. Go work on something else. I mean, go work on something that could help a lot of people. So anyway, I'm a little bit passionate about this.
No, I'm good. I'm glad to have that perspective. Well, Derya, it's been great talking to you. I really do appreciate your time and all of your insight, not only on the specific stuff that you're working on in your lab, but also in this bigger picture. And it's where can people learn more about your work? Find more about your beliefs and thoughts. And is there a website or anything that we can?
I'm very active on Twitter or X. My handle is at Derya, DERYA TR_. They can find me. I have a LinkedIn page, but I don't post much there. I have a lab page, but that's not getting updated. I do go to various podcasts and talk about these things. So if I search my name on Google, they'll probably find things. But Twitter or X, Elon Musk will get upset with me every time I say Twitter. Is probably the best way, because I post a lot about these things on a daily basis.
Thank you so much.
My pleasure. It was great.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.